Today on UPSC Science View, as science advances and DNA continues to surprise us with the amount of potential it can hold, sometimes even we don't need to look into the organism to get the eDNA, the environment holds enough traces of DNA. Sounds surprising? Hello and welcome to UPSC Science View at UPSC Practice. Today, let's learn about environmental DNA. Environmental DNA or eDNA is DNA that is collected from a variety of environmental samples such as soil, seawater, snow, or even air, rather than directly sampled from an individual organism. As various organisms interact with the environment, DNA is expelled and accumulated in their surroundings from various sources. Example sources of eDNA include, but are not limited to, feces, mucus, gametes, shredded skin, carcasses, and hair. Such samples can be analyzed by high-throughput DNA sequencing methods known as metagenomics, Metabarcoding, and single-species detection for rapid monitoring and measurement of biodiversity. In order to better differentiate between organisms within a sample, DNA metabarcoding is used, in which the sample is analyzed and uses previously studied DNA libraries, such as BLAST, to determine what organisms are present. In bioinformatics, BLAST or Basic Local Alignment Search Tool is an algorithm and program for comparing primary biological sequence information such as the amino acid sequences of proteins or the nucleotides of DNA and/or RNA sequences. eDNA metabarcoding is a novel method of assessing biodiversity wherein samples are taken from the environment via water, sediment, or air, from which DNA is extracted and then amplified using general or universal primers in polymerase chain reaction and sequenced using next-generation sequencing to generate thousands to millions of reads. From this data, species presence can be determined and overall biodiversity assessed. It is an interdisciplinary method that brings together traditional field-based ecology with in-depth molecular methods and advanced computational tools. The analysis of eDNA has great potential not only for monitoring common species but to genetically detect and identify other extant species that could influence conservation efforts. This method allows for biomonitoring without requiring collection of the living organisms, creating the ability to study organisms that are invasive, elusive, or endangered without introducing anthropogenic stress on the organism. Access to this genetic information makes a critical contribution to the understanding of population size, species distribution, and population dynamics for species not well documented. Importantly, eDNA is often more cost-effective compared to traditional sampling methods. The integrity of eDNA samples is dependent upon its preservation within the environment. Soil, permafrost, freshwater, and seawater are well-studied macro-environments from which eDNA samples have been extracted, each of which include many more conditioned sub-environments. Because of its versatility, eDNA is applied in many sub-environments such as freshwater sampling, seawater sampling, terrestrial soil sampling, aquatic soil sampling, or other environments where normal sampling procedures can become problematic. Let's take a look at the overview of environmental DNA. Environmental DNA or eDNA describes the genetic material present in environmental samples such as sediment, water, and air, including whole cells, extracellular DNA, and potentially whole organisms. eDNA can be captured from environmental samples and preserved, extracted, amplified, sequenced, and categorized based on its sequence. From this information, detection and classification of species is possible. eDNA may come from skin, mucus, saliva, sperm, secretion, eggs, feces, urine, blood, roots, leaves, fruit, pollen, and rotting bodies of larger organisms, while microorganisms may be obtained in their entirety. eDNA production is dependent on biomass, age, and feeding activity of the organism, as well as physiology, life history, and space use. Despite being a relatively new method of surveying, eDNA has already proven to have enormous potential in biological monitoring. Conventional methods for surveying richness and abundance are limited by taxonomic identification, may cause disturbance or destruction of habitat, and may rely on methods in which it is difficult to detect small or elusive species, thus making estimates for entire communities impossible. eDNA can complement these methods by targeting different species, sampling greater diversity, and increasing taxonomic resolution. Additionally, eDNA is capable of detecting rare species but not of determining population quality information such as sex ratios and body conditions, so it is ideal for supplementing traditional studies. Regardless, it has useful applications in detecting the first occurrences of invasive species, the continued presence of native species thought to be extinct or otherwise threatened, and other elusive species occurring in low densities that would be difficult to detect by traditional means. Degradation of eDNA in the environment limits the scope of eDNA studies, as often only small segments of genetic material remain, particularly in warm tropical regions. Additionally, the varying lengths of time to degradation based on environmental conditions and potential of DNA to travel throughout media such as water can affect inference of fine-scale spatiotemporal trends of species and communities. Despite these drawbacks, eDNA still has the potential to determine relative or rank abundances, as some studies have found it to correspond with biomass, though the variation inherent in environmental samples makes it difficult to quantify. While eDNA has numerous applications in conservation, monitoring, and ecosystem assessment, as well as others yet to be described. The highly variable concentrations of eDNA and potential heterogeneity through the water body makes it essential that the procedure is optimized, ideally with a pilot study for each new application to ensure that the sampling design is appropriate to detect the target. Let's learn about community DNA. While the definition of eDNA seems straightforward, the lines between different forms of DNA become blurred, particularly in comparison to community DNA, which is described as bulk organism samples. A question arises regarding whole microorganisms captured in eDNA samples. Do these organisms alter the classification of the sample to a community DNA sample? Additionally, the classification of genetic material from feces is problematic and often referred to as eDNA. Differentiation between the two is important, as community DNA indicates organismal presence at a particular time and place, while eDNA may have come from a different location from predator feces or from past presence. However, this differentiation is often impossible. However, eDNA can be loosely classified as including many sectors of DNA biodiversity research, including fecal analysis and bulk samples when they are applicable to biodiversity research and ecosystem analysis. Let's take a look at SelfDNA. The concept of SelfDNA stems from discoveries made by scientists from the University of Naples Federico II, which were reported during 2015 in the journal New Phytologist about the self-inhibitory effect of extracellular DNA in plants, but also in bacteria, fungi, algae, plants, protozoa, and insects. The environmental source of such extracellular DNA is proposed to be plant litter, but also other sources in different ecosystems and organisms, with the size of DNA fragments experimentally shown to have an inhibitory effect upon their cause-specific organisms, typically range between 200 and 500 base pairs. The self-DNA phenomenon has been postulated to drive ecological interactions and to be mechanistically mediated by damage-associated molecular patterns, or DAMPs, and to have potential for the development of biocidal applications. Let's learn about eDNA metabarcoding. By 2019, methods in eDNA research had been expanded to be able to assess whole communities from a single sample. This process involves metabarcoding, which can be precisely defined as the use of general or universal polymerase chain reaction (PCR) primers on mixed DNA samples from any origin, followed by high-throughput next-generation sequencing, or NGS, to determine the species composition of the sample. This method has been common in microbiology for years, but is only just finding its footing in assessment of macroorganisms. Ecosystem-wide applications of eDNA metabarcoding have the potential to not only describe communities and biodiversity, but also to detect interactions and functional ecology over large spatial scales, though it may be limited by false readings due to contamination or other errors. Altogether, eDNA metabarcoding increases speed, accuracy and identification over traditional barcoding and decreases cost, but needs to be standardized and unified, integrating taxonomy and molecular methods for full ecological study. eDNA metabarcoding has applications to diversity monitoring across all habitats and taxonomic groups, ancient ecosystem reconstruction, plant pollinator interactions, diet analysis, invasive species detection, pollution responses, and air quality monitoring. eDNA metabarcoding is a unique method still in development and will likely remain in flux for some time as technology advances and procedures become standardized. However, as metabarcoding is optimized and its use become more widespread, it is likely to become an essential tool for ecological monitoring and global conservation study. Let's learn about extracellular and relic DNA. Extracellular DNA, and sometimes called relic DNA, is DNA from dead microbes. Naked extracellular DNA or eDNA, most of it released by cell death, is nearly ubiquitous in the environment. Its concentration in the soil may be as high as 2 microgram per liter, and its concentration in natural aquatic environments may be as high as 88 microgram per liter. Various possible functions have been proposed for eDNA. It may be involved in horizontal gene transfer, it may provide nutrients, and it may act as a buffer to recruit or titrate ions or antibiotics. Extracellular DNA acts as a functional extracellular matrix component in the biofilms of several bacterial species. It may act as a recognition factor to regulate the attachment and dispersal of specific cell types in the biofilm. It may contribute to biofilm formation, and it may contribute to the biofilm's physical strength and resistance to biological stress. Under the name of environmental DNA, eDNA has been used in the natural sciences as a survey tool for ecology, monitoring the movements and presence of species in water, air, or on land, and assessing an area's biodiversity. The amount of relic DNA in a microbial environment is determined by inputs associated with the mortality of viable individuals with intact DNA and by losses associated with the degradation of relic DNA. If the diversity of sequences contained in the relic DNA pool is sufficiently different from that in the intact DNA pool, then the relic DNA may bias estimates of microbial diversity. Thank you very much for listening to this discussion on environmental DNA or eDNA. For more such discussions, get the UPSC Practice app from the App Store.